Good morning, guys. Can you hear me well?
Good morning.
Yes.
Thank you.
Yes.
Thank you. So it's what's Monday? Monday, November 2nd, day before the elections. And hope you had a good weekend and that you're all healthy and wearing masks. So we have one more month of Biochemistry 1, right? So we've done over 2 months of it already. So 1/3 of the class to go. Almost there. So the exam was quite a success. So I sent you some statistics. Let me know if you have any questions. And so as I mentioned to you, there will be sometime in a couple of weeks. I send you some additional problems for you to earn some extra credit, as I promised. Right. So I promised originally that I would put extra credit on the exams, but I don't know how to do it. Canvas is still a challenge for me, just like for many people. So I'll just do it the way I know how to do it.
Hey, Dr. Korienko.
Yeah.
One question I did have about the exam was, I think it's question 20, the one about nucleotides. Is like a true or false question.
Yes.
So one of the answer choices was like, oh, are nucleotides, nucleotides store genetic information? Now, like I understand, like, okay, they don't store the genetic information, they code for it, but they kind of fall underneath that. So I guess, like, I don't know, I was wondering about that one.
Well, I guess this, I know this tricked a couple of you. Yeah, so nucleotides by themselves, they do not store genetic information, right? So what's a nucleotide? A nucleotide is the nitrogenous base, ribose, and a phosphate, right? So it doesn't store genetic information, and it doesn't even code for genetic information. It's the sequence of these nucleotides, which is what codes for genetic information, right?
Yeah. I guess, like, what I was confused about was, like, as a whole, nucleotides, not a nucleotide. Like nucleotides as a whole, they code for it, and then they are part of the mechanism that stores it.
Okay, what do you refer to as a whole nucleotide?
Well, so when you say, like, so nucleotides with an S, like, multiple of them, like, so I'm thinking, like, in a chain, and like a nucleotide, like a single one is just, the nitrogenous base, the ribose, and the phosphate group. But then, like, nucleotides with an S, like, all those phosphate groups are connecting other nucleotides together,
Yeah, I think what in DNA, so we refer to this as polynucleotides.
Ah, yes. Okay.
I think it's polynucleotide which will store the genetic information.
Mm-hmm.
It's just a different molecule, right?
Yeah. Well, same molecule, just like multiple.
Yeah. So you just have to be vigilant. It's I know there are a couple of tricky questions there, but
Yeah. That one was particularly tricky.
I don't know. It was so tricky, but
I thought it was tricky. I also got that wrong. So.
Well, one question is not gonna, if you do well on this test, then one question is not gonna change things drastically for you.
It is for me. Is this closer to 87?
All right. So what do we have? So today we're gonna have on chapter 9, which is gonna have one lecture. Now this is DNA-based technologies, so this doesn't really belong in this class. So it's more molecular biology. That's why I only have one chapter, and I'll just talk about cloning primarily and protein expression, some things that are much more common with genetic engineering, and that'll be it. So and correspondingly, I will only have a couple problems on the exam regarding that. So So chapter 9 is just, we're just gonna pick a few topics, talk about it, and that'll be it. And you will learn a lot about this in biology classes. So it's molecular biology more than biochemistry. Remember, biochemistry is molecular principles behind life, right? Molecular biology is more about manipulating with large molecules and not necessarily looking at their molecular structure. All right, so molecular biology is sort of, you treat molecules like black boxes, which is not what the purpose of this class is. So we want to know what's inside this black box. We want to know what makes these molecules do or perform their function on molecular level. And so chapter 9, and doesn't really fit into our context here, but we will talk about it, just give it one lecture. All right, so let me share my screen. Let me share my screen.
I did have a question. Are we going to have homework over this chapter? Since it
Yeah.
is one.
Yeah. There is a, you have, what's total, is it total 12 homeworks, right? 12 homeworks, so you have to have a homework to earn your 10 points, right? So there will be a small homework, yeah. I will come up with some problems later today and I'll open it for you. All right. Right, so then on this slide here, I forgot to remove this. So we're not going to talk about DNA microarray technology, and we're not going to talk about DNA analysis methods. We'll just briefly discuss DNA cloning techniques and expression of recombinant proteins. If we run out of time, I still want to finish the expression of proteins. It's kind of I want to finish that and not leave it halfway there. So if we run out of time, maybe we'll spend a few minutes on Wednesday talking about expression of recombinant proteins. All right, so recombinant DNA. So basically that will be an artificial DNA that doesn't really exist in nature. Doesn't exist in nature. And we put it together ourselves. And so it forms the basis of molecular biology. All right, so here we're gonna talk about cloning of genes. Cloning of genes, you can use that to overexpress proteins, right? So produce proteins in large quantities, and also things like transgenic food, animals, to improve the agricultural products, for example. So that's all based on recombinant DNA technology. So for DNA cloning, so you have heard about this. This is on the news all the time, right? A lot of it is ethical. So scientists still debate, not just scientists, but the society as a whole debates about what's ethical, what's not. For example, creation of genetically identical copies of an organism, right? So this will be as a form of asexual reproduction, which does not involve fertilization, right? Basically, you have an egg, you remove the nucleus from the egg, you put in the nucleus from a somatic cell from a male, have it take it up that nucleus, right, and then put it into an ovum of a mother. So a lot of it is ethical. So remember, the first one that was cloned was a sheep, right, in Scotland. But now we have cloned a lot of other animals, and as far as humans, this is a huge ethical issue, right? Can we clone humans here? And one thing is important to understand, that even though that DNA may be identical, the organisms that grow up using that DNA may not totally look identical because of the environmental effects, right? So different individuals with the same genetic makeup growing in a different environment may still look different and have different different organism, different metabolism, different susceptibility to the environmental changes and so on and so forth. All right. So for organism cloning, DNA cloning, that's molecular, sometimes called molecular cloning, right? So here we're going to create identical copies of a piece of DNA or gene, for example, right? So we're going to isolate specific gene from the source organism and amplify it in a target organism. So that is done in most cases in bacteria. It's also done in yeast. So we'll actually look at some ways to do that. And so the basic steps, the way you do this is you basically cut the source DNA at the boundaries of the gene, right? So that's your gene. You cut it out here with some enzymes. We'll talk about those. And you select a suitable carrier, referred to as a vector, right? You put that in a vector, insert it, allow for the vector penetrate the whole cell and deposit the genetic information there, and let the host produce multiple copies of recombinant DNA. So let's see how this works. So this is DNA cloning, step 1. So here we're gonna generate a recombinant vector. So step 1, you know that the genetic information in bacteria, for example, is actually stored in 2 places, right? One bacterial chromosomes, and the other is plasmid DNA, right? Plasmid DNA is a circular DNA, which is much more convenient for DNA cloning for us. So what we're gonna do, we're gonna take this plasmid DNA from bacteria. We're gonna cut it with enzymes known as the restriction and the nucleases. There are virtually thousands of these enzymes that nick DNA in different positions. So for example, this yellow plasmid can be nicked at this position. And at the same time, we look at eukaryotic chromosome and we're interested in this particular piece of DNA that we want to clone. We want to clone this in bacteria. For example, E. coli, which is commonly used for this purpose. And so we're gonna also cleave this out, take this out of the chromosome using the same restriction and the nuclease, right? Same, so the restriction nuclease that you use here will be the same as is used here. And the reason for that is so that these sequences where it cleaves match the sequences in the plasmid. And if they do, then DNA ligase can actually take this sequence, attach it to the plasmid, and stitch it up and basically put it in there. And so you have the original plasmid with the target DNA incorporated into the plasmid. So we call this recombinant vector. Recombinant vector. And so the enzymes, the key enzymes that perform this for us are restriction and the nucleases, right? So they cleave DNA phosphodiester bonds at specific sequences. Very common in bacteria. So bacteria use them actually primarily to eliminate, for example, viral DNA, right? So basically they use them to recognize specific sequences in viruses that can infect bacteria, right? So viruses are not only bad for humans, they are actually bad for any cellular organisms. They thrive in host cells. And so bacteria are also susceptible to viral infections. And so they utilize restriction and the nucleases to combat to combat viruses, and they recognize specific sequences in viral DNA and cleave viral DNA using their own enzymes. Okay, so there are two major kinds of restriction endonucleases. So one type are those that generate sticky ends, and the other type are those that generate blunt ends. So on the next slide, we're gonna look at those. And there are, as I mentioned, virtually thousands of them are known. Many of them are commercially available, so you can select particular endonuclease based on your DNA sequence, based on where you want it to be cut, right? Because each of these will have a recognition sequence or will respond to a particular recognition sequence and cut in a particular site in DNA. And there is a database of all these restriction nucleases, which you can utilize to select the one that you want. All right, so let's say this is the chromosomal DNA, all right? And so this will have two cleavage sites. So this will be the recognition sequence on this end. And here, recognition sequence is CAG. Here, the recognition sequence is GAATTC. In many, so for example, for EcoR1, right? So this is the restriction endonuclease from E. coli. It specifically recognizes this sequence, which is actually palindromic. If you think about it, right? It's GAATTC, right? So this is palindromic. And so EcoR1 recognizes that. And when it cleaves DNA, it doesn't cleave it like that straight. It cleaves one strand at one position, and then it moves over 4 nucleotides and cleaves the other strand between A and G. And so it generates what's known as sticky ends. And why it is sticky is because now there are 4 nucleotides which can base pair with another strand in the process of ligation.
Is that actually palindromic? To be palindromic, wouldn't it have to be GAAAG?
Yeah, they refer to this as palindromic in the textbook. I just said it, but
I see.
But as far as the word, I myself don't really like to use palindromic with DNA, even though they use that in the textbook, because to me, the term palindromic doesn't really apply to DNA in my view. Basically, it's just it's a way to describe symmetry, I guess. It's a way to describe symmetry, and that's it. So basically, this will, in my way, this will be inverse repeat. That's how I would describe this. Inverse repeat. All right. So so this will be sticky end, right? And there is another restriction endonuclease that. So this is PVAL1 restriction endonuclease that actually generates. Oh no! Wait, sorry. This will be 2, right? So there are actually 3 kinds of now the details I don't know exactly. I think they talk about this in the textbook. We don't really need to know. I mean, as far as if you know the sticky ends and blunt ends, that's all we need to know. But so there are restriction endonucleases that can cleave the DNA in exactly the same position in both strands, right? So we call these blunt ends. And then when you have your vector, which was also treated previously, prior to this, with both restriction endonucleases, one will have a sticky end and one will have a blunt end. Then this piece here that we cut out, will go straight in there. I'll go straight in there. and the DNA ligase will stitch it up. And in general, the ligase works much better with the sticky ends. Kind of makes sense, right? So because they're sticky, so they can actually preform the double helix, and these ones require quite a bit of work because they don't really come close to, they don't really have much affinity for each other. So the enzyme has to do a lot of work to bring these blunt ends together.
So what you're saying is a little bit earlier, we don't need to know exactly how EcoRI or PVOL, split the DNA. We just need to know that there are sticky ends and blunt ends. And
yeah, I mean, that's all we need to know as far as the restriction and the nucleases. I mean, the details, you'll probably learn that in some molecular biology classes, but for the purpose of our class, that's not really important. I just want you to be familiar with this stuff. All right, DNA ligase. So these are enzymes that covalently join DNA fragments. And so obviously they are used in DNA repair. That's their major biological function, right? So if you have a cleaved DNA, so the enzymes will repair the DNA. So there's humans DNA ligases, bacterial DNA ligases. They're actually based on different principles, right? One uses ATP, the other uses NAD, NADH. All right, in step 2, okay, so once we have generated our recombinant vector, then we're going to introduce this into a host cell. So the host cell, as I mentioned to you, is a bacterial cell. And for the purposes of research, E. coli is the most common one. So we're going to introduce this vector into the bacteria. And now bacteria obviously has a lipophilic membrane. And the DNA, as you know, is highly charged, right? So the phosphate backbone is highly charged. So the DNA by itself will not get into the cell, bacterial cell, through passive diffusion. So it requires some special effort. Now there is what's known as a heat shock, where basically you rapidly change the temperature, which disrupts the membrane a bit and allows for the DNA to get in. And also electroporation. Electroporation is a technique where basically you subject the cell to an electrical voltage, a pulse, which will temporarily disrupt the membrane. And so the membrane will kind of open up and the DNA can pass through. Then you apply another pulse and more DNA will pass in, will come in, and so on and so forth. And so once you put in a certain amount of plasmids, as much as you can, then you allow for these cells to divide and propagate their plasmids, right? So the propagation of plasmids occurs separately from the propagation of the chromosomal DNA in bacteria. And so there is it's a different mechanism for it. And so the plasmid will be propagated separately. And you can see basically the cells can divide, plus in addition, the plasmids can be also multiplied within the same cell, right? So there are two mechanisms for multiplication of the genes which are part of the plasmids. All right, so the question is, how do we know? Obviously, these processes are not 100% effective, right? So some cells will take up the plasmid, some will not take up the plasmid. Some plasmids will take up the DNA, some plasmids will not take up the DNA. And so we need to select for those. And so in DNA cloning, antibiotic selection is used for that. So in most cases, penicillin and ampicillin are used, and there are a couple other, chloramphenicol, for example. So the plasmids that carry genes that oftentimes they have genes which provide the host bacterium with resistance against antibiotics. Okay, and so if the plasmid contains resistant genes, then it will produce a protein product that can be used by bacteria to destroy the antibiotic. For example, ampicillin. Ampicillin is an antibiotic, and can you tell me in this antibiotic what's the most important part of the structure that's responsible for activity of this antibiotic? Penicillin is very similar. So ampicillin is just a derivative of penicillin. So this is a type of penicillin.
Would it be the beta-lactam ring?
Yeah. So you can see this thing here, right? So this is beta-lactam. That's why they call beta-lactam antibiotics. And so this beta-lactam, if the beta-lactam is not present in the antibiotic, then the antibiotic will not work, right? It will not kill the bacteria. And so, and the enzymes which can destroy this beta-lactam ring, do you remember the name of those? Beta-lactamase. Lactamase. Yeah. Beta-lactamases are enzymes which destroy beta-lactam antibiotics. And so if your plasmid has a gene which encodes for a beta-lactamase, which will lead to the expression of beta-lactamase in the bacterial cell, then the bacteria will become resistant to beta-lactam antibiotics. So that's the principle behind protection of the bacteria from antibiotics. Okay. All right. So here, how it works with the antibiotic selection. So here's a plasmid, again from E. coli. Plasmid from E. coli. Now it actually has now two genes which confer resistance to antibiotics. Now the gene that confers resistance to ampicillin, it's a beta-lactam, right? And the gene which confers resistance to tetracycline. So here we're gonna use a different restriction endonucleases, right? So it's PST1, which also responds to a particular recognition sequence. And the recognition sequence is actually part of the gene which encodes for the ampicillin resistance gene. So in other words, when you cleave this plasmid, you cleave the ampicillin resistant gene. That's where it's gonna cleave, right? So you destroy the ampicillin resistance gene. Then you have your foreign DNA, which you'll mix this together, and they'll be ligated, specific DNA with specific DNA ligase. And so after they have been ligated, you can see what's happening here. So the tetracycline gene is intact, right? It's blue, right? And the ampicillin gene is disrupted, right? So this foreign DNA got inserted into the middle of the ampicillin resistance gene, which means that this plasmid is incapable of producing an enzyme that can confer resistance to ampicillin. All right, so so what we're gonna do then, so basically we transform the E. coli cells. So in other words, we're gonna put this plasmid through electroporation into the E. coli cells and grow them on the agar plates containing tetracycline. So what will that do? So if you grow them on agar plates containing tetracycline, so what kind of cells will survive? And what kind of cells will not survive?
The antibiotic resistance will survive.
Yeah, so the cells which have acquired the plasmid, the cells which have acquired the plasmid will survive, right? And the cells which will not, which did not, will die because tetracycline will kill them. Tetracycline will kill them. So after that, so then we will select these transformed cells. So this is agar containing tetracycline. And then but the fact that these cells incorporated the plasmid doesn't necessarily mean that they have the DNA of interest, right? So we don't know if the DNA ligase did a good job of incorporating the foreign DNA into that. And so what's going to happen is then we're going to have colonies of these. We're going to basically seed these cells onto another agar plate, okay? And subject that to two different conditions. So this will have ampicillin and tetracycline, and this will have tetracycline only. So if the agar, so the cells which contain ampicillin and tetracycline, if the ampicillin gene has been disrupted, if the ampicillin gene has been disrupted, then they will be sensitive to both. Sorry, then they will be sensitive to ampicillin, right? And so you can see here, for example, so this colony here is missing, but it's present in the agar plate, which does not have ampicillin. So if it's present here and absent here, that means that the DNA of interest has been incorporated into these cells, but not in these cells. Does this make sense? So this is used as a control. This used as a control basically to tell us that these cells have the DNA of interest. The same thing with this colony here. The second colony from the left, right? So there is a colony of cells here, but there's no colony of cells there, which means they're sensitive to ampicillin because the ampicillin gene has been disrupted by the DNA of interest. All right, so you select for the right colonies, and then so these colonies will produce the DNA of interest for you. Make sense? All right, so this is how the cloning works. Now there's so plasmids are, because they're so easy to work with and they easily get incorporated into the bacterial cells and they get replicated easily. So they represent wonderful cloning vectors for us to produce multiple copies of DNA of interest. So as we mentioned, plasmid, the circular DNA molecules that are separate from bacterial genomic DNA, replicate autonomously. They can carry antibiotic resistant genes. And so they can allow cloning of DNA up to 15,000 base pairs. Now, if you want larger pieces of DNA to be cloned, so different techniques are used. And so you can actually clone the whole chromosomes up to 300,000 base pairs. And for that, for bacteria, this technique is used as is called bacteria artificial chromosome. And you can also use yeast, right? Yeast, which is, and this technique is called yeast artificial chromosome, BAC and YAC. And so here's a cloning vector, just a bigger picture of it, right? So EcoR1, we talked about this restriction enzyme, couple others that cleave in different parts of the E. coli. Now there is an origin of replication, right? So each plasmid vector will have an origin of replication. That's where the plasmid DNA will start to be replicated. and go all the way around, right? And so, and it will have sequences which confer resistance to ampicillin and tetracycline. Now, for bacterial artificial chromosomes, BAC, so these are used again to clone larger pieces of DNA. So, so this one, for example, so this has it's CAM. This is CAM is type of antibiotic. Forgot the name of this antibiotic. Oh, chloramphenicol. Sorry, chloramphenicol. Chloramphenicol, and this is origin of replication. Now there are other genes. So these genes are actually important in the plasmid. Now think about it. So if you put a large piece of DNA into a single cell, single bacterial cell, there's a lot of possibility for transgenic effects. So in other words, the DNA can actually mix DNA from one piece of DNA sequence can undergo exchange with another one. And so these genes are responsible for keeping the content of these plasmids within the cells low, right? To basically to minimize the DNA exchange within the cell so that we don't have mutation occurring within cells. And so this gene is important for the production of light. Actually, it utilizes a dye which by itself is actually colorless, but it's an enzyme. It encodes for an enzyme known as galactosidase. So remember, galactosidases are enzymes which cleave the galactose from proteins, right? So if you cleave galactose off, then the X by itself will become blue. So X galactose galactosidase you get X, which becomes colored blue. So this is what's known as selectable marker. Selectable marker. So you can use antibiotic genes for selection, or you can use color for selecting the colonies which have the DNA incorporated into them. So this will be the bacterial artificial chromosome back vector. This is origin of replication, resistance to chloramphenicol, selectable marker for which produces the enzyme galactosidase, and the genes which are responsible for keeping the number of copies low. And so here is our DNA fragment with sticky ends. We're going to ligate it to this through the using sticky ends of the plasmid. Attach it here. We're going to use electroporation. Now this is a much larger piece of DNA, so heat shock will simply not work. You need some more powerful technique. So electroporation will actually allow for this DNA to get inside E. coli cell, right? Then you select the cells which have incorporated the DNA using by subjecting your colonies to chloramphenicol. So the cells which survive in a media containing chloramphenicol are those which contain this plasmid, this extended plasmid. And then those which have DNA in them, DNA has been incorporated and broken up the galactosidase. So if the galactosidase gene is intact, right, that means the DNA has not been incorporated. And if the gene has not been incorporated, galactosidase is active, then you get the blue color right here. So X is produced and it gives you blue color. So the colonies which are white are the colonies that you want. Does this make sense? So most of these plasmid technologies have two markers, right? One is to select for the cells which have incorporated the plasmids, and the other is to select for the cells which actually have the gene of interest. So now, as I've said to you, these processes are inefficient, and so you really need these selectable markers to guide you through the process. Does this make sense? All right, so let me see. We have yeast. I don't know if we have time for the yeast. Why don't you read about the cloning vectors in yeast? Now, this is not much different. In yeast, this will be page 327. Please read about that yourself. Page 327. Now let's talk in the remaining time briefly about gene expression in bacteria. So we can use these expression, similar techniques to express proteins of interest, right? So proteins are also isolated in small quantities, and oftentimes we want to produce gram quantities of proteins to study their function, study their structure. For example, if you want to get a nuclear magnetic resonance spectrum of a protein, you need to have a few milligrams of the protein, otherwise it's useless. If you want to use if you want to do mutagenesis, for example, if you want to mutate a particular amino acid in a protein and see what the result is, also you need to have large quantities of the protein. And so you will basically set up a gene expression assay in bacteria. So the problem is, here's the major problem. When eukaryotic genes are transcribed, you have exons and introns in the RNA, right? So bacteria cannot splice introns out of coding DNA, okay? And so what we need to do is we need to splice it before and actually isolate messenger RNA, or allow the cell to undergo splicing and then isolate messenger RNA from the cells and use that for the gene expression, right? So this messenger RNA will not have the introns in it already. So it's ready for use. It's ready for use. So remember, some of these RNAs can be monocistronic. Some of these can be polycistronic. We talked about that. And so once you have this messenger RNA, so you can't really incorporate RNA into E. coli. You have to have DNA. And so what you will do is you will synthesize what's known as a complementary DNA strand, known as a cDNA, right here. So to do that, we're going to use an enzyme. We're going to use an enzyme known as reverse transcriptase. So reverse transcriptase is an enzyme that will synthesize a DNA strand from an RNA strand. And then you get the DNA which is synthesized. Now it's a DNA where the genes are next to each other. There are no introns, all exons. And so this is the difference between complementary DNA and normal DNA. is that this DNA is much shorter and primarily has only coding regions, no non-coding regions. So this is construction of complementary DNA. So this is the messenger RNA. We're gonna add the primer. We're gonna add the enzyme reverse transcriptase, add the deoxynucleotide triphosphates, make the DNA in the 3' direction, right? So the blue one will be the DNA. The original one will be the RNA. It's a hybrid. And then we take advantage of the fact that RNA is susceptible to basic conditions. We can degrade it easily, right? We can subject this to higher pH values, and the RNA will be degraded, and this DNA will remain intact. Again, we add a primer and synthesize duplex DNA. Synthesize duplex DNA, and now we can use this duplex DNA to insert this into the plasmid, right? Insert this into the plasmid. And so these plasmids will be called expression vectors. And they have a lot of interesting features, many more features than cloning vectors, right? So they'll have promoter sequences, operator sequences, code for ribosome binding site, transcription termination sequences. So this will be typical expression vector. So there is a promoter and operator sequences. Operator is used primarily for regulation. This is where the messenger RNA will start to be produced at the promoter region. Linker, transcription termination sequence. Now you also need a selectable genetic marker for antibiotic resistance, origin of replication. So these actually expression vectors can be very different. They have a lot of different features. They differ from each other by many different features, and they're actually they're constructed depending on which organism you use for expressing the proteins. And so once you have expressed particular protein, then you have to purify your protein, right? Remember, we talked about protein purification a little bit. We talked about gel electrophoresis and things like that. Chromatography, right? So the advantage of making a protein is that you can attach a sequence to this protein, which will allow you for easy purification. For example, something which is called as histag. Histag, is anybody familiar with histags? So your protein will contain a histidine sequences which will bind to nickel, and you can basically pull down those proteins with nickel. Now there are, you can also attach a sequence to your protein which contains the enzyme glutathione S-transferase. Glutathione S-transferase will have binding affinity for glutathione. And if we look at the next page, so this will be the chromatographic purification. So this will be affinity chromatography. We talked about that before, affinity chromatography. So here, for example, this is the gene for the target protein, red, right? And we're gonna actually make a fusion protein. We're gonna use the protein, the target protein that we want, and we're gonna fuse it with glutathione S-transferase. And we're gonna transcribe this in the bacteria, right? This will be the fusion protein. The red one is what we want. This color, I don't know what this color is there for purification. we're going to express this fusion protein in a cell, right? Prepare cell extract containing the fusion protein, part of the cell protein mixture. And so this column will have the beads that have glutathione attached to them. And you can see these little spheres here with the little mouths. These little mouths will have affinity for these spheres of the which is actually glutathione. So if this is glutathione S transferase, this is glutathione itself. So the enzyme will have affinity for the ligand. And so when you attach this to the apply this to the column, then all the other proteins will come through, but the proteins which have glutathione as transferase as part of the fusion protein, they will stick. They will stick and they will not go through. And then after that, after you got rid of all the non-specific proteins, then you apply a solution of glutathione, which is free. You add it to the column, and it will compete with the beads, right? Compete with the beads and wash the whole fusion protein down. And the fusion protein is eluted by glutathione solution. Now, the trick, of course, is that at the end you get a fusion protein, not just a protein of interest. You have to find an enzyme, a protease, which will allow you to cleave the fusion protein at the site where you want, right? So there must be a recognition sequence here for a protease, which will allow you to get rid of the GST. All right, any questions? You know, I went a bit too fast, but this is really not such a difficult material, right? As I mentioned to you, it's mostly biology. There are a couple of the slides that I have here that I think would be good for you to know. So for site-directed mutagenesis, I think there is one page specifically figure. So this is page 331. Please look at that. And qPCR, a very cool new application for polymerase chain reaction. I want you to look at that as well. This will be page 334. Page 334. All right, do you have any questions, guys? No? So how's this material? How does this compare to what we normally talk about? Which one do you like better? Do you like molecules better, or do you like working with bacteria and cloning DNA and not really knowing what the molecular structure of the species are? You know, it's like, you know, what's this expression? What do you say that lack of knowledge is bliss? How does expression go?
Ignorance is bliss.
Ignorance, there we go. Ignorance is bliss, right? So it's easier to live and have a stress-free life if you don't know what these things consist of, right? But it limits you. It limits you in what you can do. If you do know the molecular structure of the molecule that you're working with, it gives you some extra capability to manipulate this and develop new techniques and turn you into better researcher or better doctor, better scientist, better anybody. So
Those extra credit questions you're talking about, are those going to be on Canvas or on Sapling? And when will those be available?
yeah, today. So I will probably later this morning.
Okay, cool. Thank you.
All right, guys. See you on Wednesday. On Wednesday
When
we'll
are
have
those extra credit questions due?
extra create questions. Well, I still haven't made them, so sometime in November. Definitely before Thanksgiving.
Okay, thank you.
Yeah. Bye, guys.
Thank you.
Mhm.